All right, so I had made this PowerPoint to go over with you guys in class on Monday, but then I had that Embry meeting, and so instead we're just going to have this recorded PowerPoint lecture situation. Okay, so you guys already know about transcription and translation. The main point of today's presentation is to discover how the RNA polymerase knows where in the DNA to attach, and then how the ribosome knows where on the messenger RNA to attach. Before we get into that, though, there was a couple of other things that I wanted to make sure that you are familiar with. They're kind of like lingo in genetics, and I want to make sure that you know them. First is gene expression. So gene expression is making messenger RNA and then making proteins. So if a gene is expressed, then you will be able to find messenger RNA and protein in the cell. Okay, so gene expression is making protein. There are pieces of DNA that are called open reading frames, or ORFs, ORFs, ORFs. Open reading frames are pieces of DNA that have start and stop codons on them, but we do not know if these pieces of DNA are expressed into proteins or not, because we have not found the protein. So ORFs are pieces of DNA with start and stop codons, which may or may not be expressed as proteins. So ORF is a more general word that you can use and that you'll see used a lot in biology in case you find a piece of DNA that looks like a gene but is not made into a protein. So that's what that means, is just that we have not been able, maybe we have not been able to recover the protein. Maybe we have, and we're still using the word ORF. ORF is just a broader classification than gene. Genes specifically mean that we have found the protein and we can point to it. ORF mean that the piece of DNA has a start and stop codon and should make a protein, but we may or may not have found the protein. Okay? Okay, I found this genome map of E. coli and wanted to show it to you guys. So you remember that all DNA in bacteria goes into a circle, right? So the bacterial chromosome is circular. The first step when the phage DNA enters the host cell is for the phage DNA to circularize. So bacterial DNA is in a circle. And so that is what we can see here is this circular chromosome map. The origin and the terminus have to do with copying the DNA. So you do not need to know about those right now. Do you remember when we talked about how all DNA, when we talked about how all RNA comes from DNA? So here you can see the tRNA and the rRNA genes. So these genes are not as common, not as widespread in the chromosome because they just have one job. So we don't need as many different versions of them, but they are still in the chromosome. So all RNA in the cell is encoded by the DNA in the chromosome. There's not like some backup RNA stash for the tRNA and the rRNA genes. All RNA in the cell comes from DNA. So this gold up here is representing genes that are on the top or forward strand of the DNA. And so right here where, my mouse is not very cooperative, right here where this piece is kind of sticking up, this is a protein coding gene. And then this yellow color are genes on the reverse strand. We'll get to this a little bit later today and then again on a different day. But what I really want you guys to see here, besides the tRNA genes and the rRNA genes, is the density of protein coding genes in the chromosome. And so this really makes us stop and ask, where does the RNA polymerase go in order to make a messenger RNA transcript, and how does the RNA polymerase, quote unquote, know where it should be going? So that's what we're going to learn about today. When you guys first learned about transcription and translation, you probably learned about a single piece of DNA where the whole thing became the protein. So you would have just learned about the coding region. So you would have learned the start codon is up here, the stop codon is down here. This whole thing is going to be read and expressed as the protein. In the actual chromosome, there is a bunch of flanking DNA. So flanking means on the sides, like the flanks of the horse is like the sides of the horse. So flanking DNA is DNA on the sides. And in the chromosome, you have the coding region, which will become the protein. But then on either side of the coding region, you have some regulatory sequences that do not become the protein, but those regulatory sequences help to determine when the protein is made. And so those are the sequences that we're going to be talking about today. So the coding region here in the middle is going to be the region that becomes the protein, indicated by this red dashed line. Then just out from that will be the mRNA. So the mRNA is going to include the leader all the way to the end of the trailer. So the mRNA is made by the RNA polymerase, as you know. So if the mRNA starts here, then the RNA polymerase must attach at the promoter, because the RNA polymerase has to be on the DNA in order to start making RNA. So sure enough, the RNA polymerase attaches to the DNA at the promoter, and the RNA polymerase will be reading the template strand, okay? So the RNA polymerase will bind and will be reading the template strand. So the RNA polymerase reads the template strand and then begins making messenger RNA immediately after the promoter. Binds at the promoter, starts making mRNA right after the promoter. Makes mRNA this whole way until the end of the trailer, and then the RNA polymerase comes off at the terminator. And so you can tell where the RNA polymerase and the ribosome come onto the DNA/RNA and where they come off by looking at the piece that is made, because the piece that is made has to be while the RNA polymerase is on the DNA. So the RNA polymerase comes on at the promoter, reads and transcribes all of this, and then comes off at the terminator. Okay? The ribosome is going to come onto the messenger RNA here in the leader region. We will learn the name for the specific place where it binds in a few slides. But the ribosome will come on at the leader, and then it will make the protein, and then it will come off at the trailer. And again, we know that because the protein can only be made while the ribosome is on the messenger RNA. And since the protein is made in the coding region, the ribosome must come on at the leader, and the ribosome must come off at the trailer. You may remember that in bacteria, because there's no nuclear membrane, transcription and translation happen simultaneously. So here is the RNA polymerase. The promoter was somewhere up here. So the RNA polymerase bound at the promoter, started reading the DNA and making RNA. As soon as the leader is produced, then the ribosome will attach to the leader and begin making protein. So that's the way that works. Okay, so RNA polymerase binds at the promoter. Okay, so this slide shows you an electron micrograph of phage del Rio, which is one of our Gordonia terrae phages from last year in Phage Lab. You guys already have seen pictures like this. You immediately know what phage family this is in. Look at the long and flexible tail. Okay, so we've got the capsid here, and then we've got the phage tail. So what I want to remind you guys on this slide is that the virion, remember the name of the extracellular or the virus particle outside the cell is the virion. The virion is made up of the structural proteins. Okay, structural proteins. So this diagram here shows you some of the different structural proteins involved in making the virion. So the capsid is kind of built like a, the things that you like hit with sticks and candy comes out, a piñata. The capsid is kind of built like a piñata. And so there are these scaffold proteins, these little purple ones here, that go in, and then the major capsid protein is built around the outside. So if you've ever made a piñata, this is kind of like the balloon and the paper mache part. So the scaffold proteins are the support inside, then the major capsid protein is built around the outside, and then the portal is kind of like a doorway into the capsid. So once the major capsid protein is all assembled, then the scaffold protein is taken out by proteases. Proteases are enzymes that break down other proteins. So the scaffold protein is taken out, and then terminase is used to stuff the DNA inside of the capsid, so you get this. Then there's this other little structural protein called the head-to-tail connector, and it functions kind of like the neck of the phage. It's where the tail will attach. And so then, about the same time, the tail is being built on its own. And so in the center of the tail is called the tape measure protein. We'll get back to this, but the tape measure protein runs the whole length of the middle of the tail and determines how long the tail will be. So you have the tape measure protein, and then the different major and minor tail proteins are built around the outside of the tape measure protein with help from the tail chaperones. So that's why the tape measure protein determines the length of the tail, because it's kind of like the scaffold around which the tail is built. And so then we get the tail tip added, some additional tail proteins are put on, and then the tail is attached to the head-to-tail connectors. So those are the structural genes. Do you guys remember when we, we'll get to this in a second. Do you guys remember when we talked about early and late genes, that the early genes are genes having to do with the DNA of the virus or with the initial infection process? So things like integrase, the repressor gene, those are all going to be early genes. The phage needs those genes as soon as it gets into the host cell. Whereas the late genes are genes that the phage needs expressed immediately before it lyses the host cell. So the late genes are all going to be the structural genes, the genes that encode the structural proteins for the virus, the major capsid, the tape measure, the tail proteins, all that stuff. Those are going to be structural proteins that will be expressed late. Keep that in mind. One other term that I need you guys to know is this term polycistronic. Okay, so a cistron, right here, C-I-S-T-R-O-N, is an old-timey word for gene. And so each cistron encodes one protein. So in eukaryotes, each gene gives you one messenger RNA, gives you one protein. It's really straightforward. In bacteria, it's a little bit different. So bacteria have this awesome feature where they can make polycistronic messenger RNAs to where multiple genes will be represented. So poly is many. Cistron, remember, is an old word for gene. So each cistron can make one protein. So polycistronic messenger RNAs are messenger RNAs that will give you multiple proteins. And if you look up here, you will see that this piece of DNA has a single promoter. And you'll remember the promoter is where the RNA polymerase attaches. Okay? So you're like, why do bacteria do this? This seems very weird. This is actually super handy. So bacteria will often need multiple proteins at the same time. A great example of this are the phage structural proteins. So all of those proteins that are needed to build the capsid and to build the tail are needed at the same time at the end of the infection process. So wouldn't it be super handy if those were all turned on by the same promoter? If one single signal would tell the RNA polymerase bind here and make the RNA for all of these structural proteins at the same time because we need them together. That is exactly what happens, and that gives you a polycistronic messenger RNA. So this might be like the major capsid protein, the scaffold protein, the portal protein, maybe the tail proteins are over here. So when the bacterial cell, and by extension, when the phage needs a bunch of proteins at the same point in the infection process or at the same point in the life cycle, then they will put all of those genes after one single promoter. So the RNA polymerase binds to that promoter, and then it will make the DNA, it will read the DNA and make messenger RNA for all of the proteins in a single gigantic piece of RNA. And then the ribosome will come on, make this protein come off, come on, make this protein come off, come on, make this protein come off, until all of the proteins have been made, and those are going to be proteins that are needed at the same time. Okay. So this is what we're going to be starting on next week. This is a little bit of a snapshot. I'm very excited. So up here at the top, weensy and unreadable, is the entire phage genome map for phage del Rio. So each of these little boxes is one gene. Remember how on the E. coli genome map there was like the genes on the forward strand of DNA and the genes on the reverse strand of DNA? So all of the boxes that are on top of the line are the forward genes, and these boxes here that are underneath the line are the reverse genes. Okay, forward genes are read like from right to left. That's what makes them forward. Reverse genes are read from right to, wait a minute, I think I said that wrong. Forward genes are read from left to right, like normal reading. And then reverse genes are read from right to left. Okay. So what I want you guys to see here in this blown up version, this little blown up part here is, do you see this blue line? So I've taken this blue line section and blown it up here. And if you read the names of these genes, you'll be like, wait a minute. Those are the structural genes we just learned about. So here is the scaffold protein gene. Here is the major capsid protein. Here is the capsid maturation protease, which is this little green Pac-Man dude here. Here is the portal protein, which is the doorway into the capsid. Here's the two pieces of the terminase, which are what stuff the DNA in. There is a bajillion head-to-tail connector proteins right here. And then we have major tail protein, minor tail protein, tail assembly chaperone proteins. Here is the tape measure protein, which is kind of the scaffold for the tail. Then there's some minor tail proteins and other tail proteins over here. Okay, so all of these are the structural phage proteins. When does the phage need these? Early or late? The phage needs these late, and it needs these all at the same time. And so all of these are going to have a single promoter. And so the promoter is probably right here. So the RNA polymerase will attach here, and then it's going to read all of this DNA and make a gargantuan messenger RNA that is polycistronic, contains all of these individual genes within it. Okay. Hopefully that makes sense to you guys. The next thing I want to focus on is this little section right here. Measure protein. It is always the longest protein in the genome. You can see it right here. When in doubt, just guess it's always the longest one, always. I guess that's not really guessing. Anyway, so there is a single protein called the tape measure protein, which runs the whole length of the tail right here. And then the tail is built around that. Then the tape measure protein comes out, and it is that gap that makes the channel where the DNA goes through to infect the host. It's kind of like a straw at that point. Okay, so that's the tape measure protein. Goes inside of the tail and then comes out and leaves a gap. Okay, the reason I showed you that is because I wanted to show you this. So this sequence right here are the first 20 amino acids of the Del Rio tape measure protein, where each amino acid is represented by its one-letter abbreviation. I know that you do not know all of these amino acids, but I'm likewise pretty confident that you know M is methionine, mostly because that's the only M amino acid. So here we've got one methionine. We're like, of course, because methionine is the start codon, the first one is methionine, obviously. But we keep going down here and we see, wait a minute, a whole nother methionine, not the start codon. So it is true that the start codon starts the protein, but it's also true that the start codon codes for an amino acid. Its whole job is not just to start the protein. Its job is actually to code for an amino acid. And so how does the ribosome tell which start codon is the start codon and which start codon is just a random other AUG that will code for methionine? So do you remember when we were looking at that long sequence that had the coding strand and then the promoter was up here and then the leader was in there? Within that leader region is where the ribosome attaches, and very specifically, the ribosome attaches to something called the Shine-Dalgarno sequence. Okay, this is named for the two people that discovered it, Shine and Dalgarno. It's a man and a woman. I think that they're British or Australian. They're not from the US. Anyway, the important part of this is that these are their names, so you may not misspell them and you must capitalize them. This is important. Okay, so the Shine-Dalgarno sequence is immediately upstream of the start codon. So here is the start codon, here is the Shine-Dalgarno sequence, and you will see that the Shine-Dalgarno sequence kind of hooks around the front of the small subunit of the ribosome, and the Shine-Dalgarno sequence is in the messenger RNA, and the Shine-Dalgarno sequence in the messenger RNA physically binds to the ribosomal RNA. And so the RNA on both accounts is very important here in figuring out where the start of the protein will be. So the RNA polymerase attached at the promoter in the DNA. The ribosome attaches at the Shine-Dalgarno sequence in the messenger RNA. Okay, ribosome attaches at the Shine-Dalgarno sequence in the messenger RNA. And it's the Shine-Dalgarno attachment that determines which AUG actually is the start codon. AUG by itself is not descriptive of the start because you can find methionines anywhere. It is the Shine-Dalgarno sequence and its interaction here with the small subunit that really determines where the start of the protein will be. Okay, so what I want you guys to see here is all of these are the start codons. We'll get back to GUG in a second. When we look for the Shine-Dalgarno sequence, we are looking for something that is high in A's and G's. A's and G's are called purines. And the Shine-Dalgarno sequence is characteristically high in A's and G's. It's going to be about 10 bases upstream of the start. It can be like 8 to 13, but it's about 10 bases upstream of the start, and it's 3 to 10 bases long, and it's got GGA, GG, or something relatively like that in the middle. Okay, so here we've got GGAG is the core. Here we've got that. Here we've got GGGU, GG. Here we've got AGGA. So it's not always going to be exactly GGAGG, but the core will have a concentration of A's and G's will be 10-ish bases up from the start codon. And it is, again, this Shine-Dalgarno sequence is where the ribosome binds, and thus what determines the start of the protein. The Shine-Dalgarno sequence is also sometimes called the ribosome binding site or the ribosome binding sequence or the RBS. When you take genetics, this will make more sense. Okay, so here is what I want to show you guys. So this is the 6-frame view of the DNA, right? So here's the top strand of the DNA, bottom strand of the DNA, the 6 different amino acids that can be made at any given point. So here is the methionine that starts the tape measure protein. This is another protein. So here is, so we're in the coding strand, right? Because it looks like the RNA except no U's. So that's the coding strand of DNA. It's the strand that looks like the RNA. So here is the start codon in the coding strand. And then, so we're going to count up like 10-ish bases. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And then I'm going to start to look after that. I've lost the mouse. It's somewhere here. Okay, so I think I was here. And I don't see there's no G here, but so it's 10-ish bases, right? So at the 11th base, I start to see GGAAG. And doesn't that look really similar to GGAGG? So this is the ribosome binding site right here. So this is actually what the ribosome attaches to determine that this is the start codon and that this is not the start codon. Okay. So next week we're going to learn about this chart thingy. But I wanted to go ahead and show you when you are choosing. So part of what you'll do later on this semester and in bioinformatics next semester is choosing which of the starts is the correct start. And so you'll look at a combination of factors. One of the things you look at is length of the gene or open reading frame. Remember, that's what we call it if we're not sure it's making a protein. So you'll consider open reading frame length, but you also consider Shine-Dalgarno score, because the more likely the ribosome is to bind, the more likely a protein is made there. Okay? And so less negative scores are better. And so here is this particular start for this tape measure gene. Here's the ATG at 12,041 base pairs. Here is the GGAAG that we found up here. And the score for this Shine-Dalgarno site is minus 3.6. Okay, minus 3.6 is like about 1,000 times better than minus 5.9. So minus 3, the smaller the number, the better. So minus 3 is better than minus 4, is better than minus 5, etc. It's kind of like exponents. Because minus 3 is 10 times better than minus 4, which is 10 times better than minus 5. And since this is almost minus 6, then this one is going to be about 1,000 times better. So we would choose this to be our start. We'll go back over this later, but this is how we use the Shine-Dalgarno sequence, because this is how the cell uses the Shine-Dalgarno sequence. The ribosome will bind to the most preferred binding site. Okay, so you might have noticed all of these other random start codons. What is going on with that? So we've got the AUGs. Remember that when people write down genes, they normally write down the coding strand. They don't usually write down the RNA. And so it will say ATG, but it's the same as the RNA. So this is going to be the methionine codon here, AUG. It's just written as the coding strand. Bacteria can use alternate start codons. So eukaryotes mostly only use AUG. Bacteria can use GUG, which in the coding sequence is going to be, or on the coding strand is going to be written as GTG. And then bacteria can also use TTG, which in the messenger RNA will be UUG. So do be aware that although eukaryotes only use AUG. Bacteria use AUG, GUG, and also can use UUG. And since the phage are parasitizing the bacteria, then the phage use whatever the bacteria use. Okay. So just to recap, this is the entire section of DNA. The coding region is flanked by these regulatory pieces. The RNA polymerase attaches at the promoter. The ribosome attaches at the Shine-Dalgarno site, which is within the leader here. And then we'll get transcription and translation, aka gene expression, and then we will end up with the protein.